Hello everyone, welcome back to my channel, igcse biology help. In today's video, I'm going to be solving the biology paper 4, the variant 2. This is from the November 2022 series. I have already posted the variant 1 exam. You can find it on my channel. Make sure to subscribe to my channel and like my videos so you can receive more content like this. Let's start solving. Question number 1 says phytoplankton consists of many species of single-celled and many-celled algae. Algae are classified in the protist kingdom. All algae contain one or more chloroplasts. State the name of another kingdom that contains organisms which have chloroplasts. Of course, we all know that chloroplasts are found in only plants. You cannot find them in animal cells. Okay, so the second part says a student investigated the species composition of the phytoplankton in a lake. Figure 1.1 shows some of the phytoplanktons collected by the student. The actual length of algae X is 0.19 millimeters. Convert the actual length of algae X to micrometers. So we know that 1 millimeter equals to 1000 micrometers. So this is the conversion you're going to be using. You're going to be converting from 0.19 millimeters to micrometers by multiplying by 1000. You're going to get 190 micrometers, so that is the answer. C says the student made careful drawings of 7 types of algae found in the samples of lake water. The drawings are shown in figure 1.2. And use the key to identify each of the species A to G. Write the letter of each species in the correct box beside the key. So over here we can see that in 1 it says algae has 4 spine-like extensions. So this is actually found in F, and it's also found in G. We can see that there is 4 extensions coming out of the organism, and we can also find this in F and G only. So let's see. It says go to 2, so we're over here at 2. It says that algae has spine-like extensions that are all the same length. So we will check which one has the same length, or approximately the same length. We are choosing between F and G because these are the only ones that have extensions. We can see that G has approximately the same length of the extensions. Over here in F, there is this one tall extension, and then there's shorter extensions down below here. So our answer for this one is going to be G. So for the second one in 2, it says algae has spine-like extensions that are different length. This is the F, because as I just said, it has different length. Over here, this one is really tall and this one is shorter, and this one is like a little bit shorter, and so our answer is going to be F for this one in number 2. So number 2 is done. Let's go to 3. 3 over here in number 1, we're going to go back. It says algae does not have 4 spine-like extensions. So this is basically like E and C and D and A and B, like all of them, they don't have extensions. So let's see, and it says go to 3. So over here in 3, it says algae has a round shape. So actually this is going to be, the round shape is actually going to be B. We can see that it's like a circle over here, which is circular. So actually our answer is going to be B. So this one says algae does not have a round shape. This is it says go to 4. We can see that right now we have finished B and we have finished F and G. So we have E and we have D and C and A left. So it says go to 4. In 4, it says algae narrows towards the ends. It says go to 5. So it says algal cell is semicircular in shape. This is actually D. We can see that it's a semicircle. Semicircle means a half a circle. So this is actually D. So our answer is going to be D for 5A over here. Now for 5B, it says algal cell is not semicircular in shape. This is actually going to be C because it is like this shape. So we only have A and E left. It says go to 6. it says in this one in 4b, it says alga does not narrow towards the ends. This is actually A and E. We only have those left. C is done. So A, it's not narrowing towards the end, and also E, over here, this one, it's like a straight like stick. So in 6 says the alga is branched. So actually this is A. We can see that it's branched because it has like several branches, just like a tree. So this is A. Now on the last one, of course, it says the alga is not branched. This is going to be E. So this is how you solve this question. D says phytoplanktons are photosynthetic organisms. Describe the importance of photoplankton in food web of lake ecosystems. So first of all, photosynthetic organisms, they will provide energy for the whole web. This is because they will create energy or glucose from the sunlight and from the water and the carbon dioxide, and this will provide energy to start off the food chain. So first of all, phytoplanktons are producers. They convert sunlight to chemical energy, which is glucose, and this can be used when other animals will eat the plant. They will gain energy, and so the energy will move on from one consumer to the other. This is because the producer has started this energy chain, and it's a source of food for all trophic levels. Phytoplanktons convert water and carbon dioxide to glucose energy. So you're going to get a mark if you describe the method, the photosynthesis method that it will produce energy from, and you're going to say that phytoplanktons are producers. And what does the producer do? It provides a source of energy for all trophic levels. 2 says figure 2.1 is a diagram of the human alimentary canal and associated organs. Table 2.1 shows enzymes, the organs that secrete these enzymes and their substrates and products. Complete table 2.1. So over here we can see that in this one, the first one, it says the enzyme is amylase. We all know that amylase is secreted from the salivary gland, and actually the amylase, its only job is to break down starch and produce from that maltose. So the second one is protease, or you can say pepsin. It is present in the stomach, and we can see in number 3 over here, this is where the pepsin is produced, or where it is present. And in number 1, this is actually the salivary glands, where there is amylase enzyme produced. Now it breaks down the protein, of course, and produces amino acids. Now for lipase, this is the enzyme that breaks down fat and oils, and it's released from the pancreas. You can see that this one that I have highlighted, this is the pancreas at number 4. And so it will produce, after breaking down the fat and oils, it will produce fatty acids and glycerol. So maltase, it is produced in the small intestine in number 5. You're supposed to fill this one in. This is the small intestine, and this is the large intestine. So it will break down maltose, and it will produce glucose from this one. So basically it will take down the maltose that is produced by the amylase, and it will produce glucose. B says, explain the role of hydrochloric acid in the alimentary canal. So we all know that hydrochloric acid is present in the stomach, and this enzyme is present so that if someone ate contaminated food that contains bacteria and harmful viruses, this hydrochloric acid, it will break down or denature the harmful pathogens enzymes. It will make their enzymes not work as efficiently. Denaturing will occur by changing the active site shape. This will not let the pathogens to reproduce or do their reactions, because it is already damaged. So it will also provide optimum temperature or conditions for enzyme pepsin. So we all know that the HCl is very acidic, and the pepsin works in pH 2, and this is the optimum temperature. So HCl provides the optimum conditions for that enzyme. C says, state a function of the region of the alimentary canal labeled 6 in figure 2.1. So over here we can see that at number 6, this is actually the large intestine. I have just said that in the previous questions. So we all know that the large intestine, what it does is it reabsorbs water and ions And so this is where the stool comes out after the digestion. D says describe how food eaten by humans is reduced to smaller pieces and explain how this makes chemical digestion more. So first of all, we're going to say that the food is reduced by mechanical or physical digestion. This is done by the mouth. This is done by chewing and grinding food. The chewing is done by the teeth in the mouth. Specific teeth do the cutting, which is the incisor, and the premolar. Actually, as we all know, you can also say the premolar and molars, or just the premolar. You can say that it crushes food. The premolars are the back teeth in your mouth, the thick teeth that you have. Also, the food is mixed with saliva. This is to help to help the food be soluble. Emulsification is done by bile, occurs in the small intestine. Emulsification is the breaking down of like food molecules by the bile. It breaks down the large fat droplets into smaller droplets. So this is like the enzyme lipase. It breaks down the fat droplets or the oils. So this is the answer of D. Aphids are insects that feed on phloem sap by inserting their mouth parts into the phloem tissue. Figure 3.1 shows an aphid feeding on the phloem tissue in a leaf. AI says state two features visible in figure 3.1 that identify this animal as an insect. So over here we can see that all insects have thorax, head, and abdomen. But if you're going to say two features of insects, make sure that the feature that you are saying, it is actually like visible in the area or the visible in the picture that is shown over here. So for example, you don't say exoskeleton because we can't see the exoskeleton, for example. So over here we can see that there's three body segments: thorax, head, and abdomen. The thorax is basically the area in the middle, and the abdomen is this area at the end, and the head of course is over here. And there's three pairs of jointed legs. We can see three pairs means that there is 6 legs in total, and we can see that of course over here. You can see that there's 3 feet or 3 legs over here, and the other 3 are on the other side. Double I says aphids can transmit viral pathogens when they feed on the plant. Suggest how a severe infestation of aphids on the crop plants can be avoided. So first of all, you can say that you can use pesticides or insecticide. These will basically act like the bug spray. When you spray bug spray, you repel away the bugs. So this is basically like that. You can also say that you can cover the crops, like for example, you don't grow the crops out in like an open area like a garden or something where it can be like where there can be flies that can enter. So you can basically cover, or you can also use like genetically modified crops that are resistant to insects. Genetically modifying the plants will also help in the longevity of the plants. So this is a way, but it's very expensive to do this method. Triple I, the aphid shown in figure 3.1 is feeding on a phloem tissue in a leaf. State the names of 2 tissues in the leaf that the mouth parts of the aphid passes through to reach the phloem. So you will basically say any tissue or any structure inside the leaf. You can say palisade mucophyll cells, you can say spongy mucophyll cell, you can say the upper epidermis, you can say anything you want. It is valid in this question. B says aphids have been used to investigate the translocation of sucrose in the phloem tissue. While they are feeding on the phloem sap, aphids excrete a sucrose-rich fluid known as honeydew. In an investigation, 2 groups of 4 aphids were placed at intervals along the stem of a young willow plant, as shown in figure 3.2, and the leaves were enclosed in an airtight glass chamber. A special form of carbon dioxide gas that contained radioactive carbon-14 was supplied to the leaves for a short period of time. Samples of honeydew were collected at intervals from the groups of aphids. The time taken for sucrose containing C14 to travel the distance between group A and group B was recorded. The investigation was repeated twice using a fresh stem and different groups of aphids for each trial. The results are shown in table 3.1. I says complete table 3.1 by calculating the rate of movement. So to calculate this rate of movement of the C14 in trial 3, you're going to be converting the minutes that you have in the time taken to hours. This is by dividing the amount of minutes you have over 60, because in 1 hour we have 60 minutes. For example, you're going to get 2 because there is 120 minutes in 2 hours. So you're going to be dividing 650 over 2 to get your rate of movement per hour. Now you're going to be repeating the same thing for 2. You're going to be dividing 75 over 60. You're going to get 1.25 hours. You're going to divide the 340 over that number of hours so you can get the rate. And you're going to repeat the same thing for 3. Eventually, you're going to get, if you divide 630 over 2.5 hours, you're going to get 252 millimeters per hour. Double I says, outline how C14 and carbon dioxide gas becomes more incorporated into the sucrose molecules that are translocated in the phloem. So we know that it's basically asking how do the carbon dioxide gas enters into the like the sucrose molecules inside the leaf. You're going to say, first of all, we know that carbon dioxide enters to the leaf through the stomata as a raw material for photosynthesis. Carbon dioxide is used and needed for photosynthesis so that the plant can produce its own energy source, its own glucose. And so the photosynthesis occurs in the chloroplast, and the glucose is converted to sucrose for storage. You can also say that it will be turned into starch if you want, but you will generally get more than the full mark if you write all of these points. CI says sucrose travels from the stem to other parts of the plant known as sinks where it is used. State 2 parts of a plant that are sinks for sucrose. So you can say root tips, you can say all the list of answers that I've written down here. You can say flowers, fruits, seeds, and you can say organs, young or growing leaves. All of these like structures, they need glucose so that they can like grow. So this is what is a sink. Double I, sucrose is used in the cells of the sinks in a plant. Describe the uses of sucrose by sinks. So you can say that first of all, sucrose is converted to glucose, and the glucose is respired by aerobic respiration to provide energy for growth and reproduction and all of the like stuff that is written down. You can say any one of these, and you can also say that it is stored as sucrose or starch. After like if a plant has starch, it will break it down first of all, and after that, if there is excess glucose that it doesn't need, it will turn it back into starch so it can be stored for when the plant will need it. So it is also used to make cellulose for cell walls. We all know that the cell walls of the plants are made of cellulose, so this is a use for sucrose. And you can say that it's converted to amino acid to make proteins. For a figure 4.1 is a flow chart showing some of the processes that occur in a biofuels power plant. The fermentation stage shown in figure 4.1 requires yeast. Complete the balanced chemical equation to show how ethanol is produced by yeast respiration. So if you took chemistry, then you know about this fermentation process and its advantages and disadvantages. But fermentation is actually anaerobic respiration basically. So you're going to write down the equation of anaerobic respiration, which is this is glucose, and this is ethanol or alcohol, and this is carbon dioxide. You have to balance it out. And yes, so this is how you solve this question. Double I says, using the information in figure 4.1, suggest the environmental advantages of using ethanol as a fuel. So ethanol, of course, is a renewable resource. It is not like fossil fuels. So ethanol has a big advantage that it will reduce waste from forest, or it will overall reduce waste. It will also reduce the amount of carbon dioxide that is produced into the air. And so when there is less carbon dioxide in the air, there will be less global warming and there is less pollution overall. And it is also going to cause less harm to the environment from mining or drilling, as we all know that if you want to get fossil fuel, there has to be a lot of mining and drilling. And it's also a way to reduce the use of fossil fuels and renewable resources such as fossil fuels and coal. Um, and of course, as I said, it has no carbon print, which means it does not produce carbon dioxide when it is burned or used. Triple I, farmers grow giant reed plants as monocultures, describe the disadvantages of growing giant reed plants to provide biomass for the production of biofuels. So, monoculture means that, for example, if a farmer has a land, um, he or she will grow only, for example, apples, for example. He or she will not grow any other type of fruit or any other type of crops. So this is what is monoculture. So of course this can have several disadvantages. For example, if there's a plague or if there's an like viruses, if it spreads in one plant, then the whole farm will be infected and the whole yield of the crops will be lost or it will be damaged. So this is the first disadvantage. Also, the land can be used in other ways such as food productions, and also you can say that there is high input of pesticides, herbicides, and water needed. This is more than usual because this is monoculture and there is higher risk of viruses and plagues and pathogens. Also, there is loss of habitats, which cause loss of biodiversity and genetic diversity. So there is many things you can write in this question, and it's also 2 marks. So what I've written down over here will get you more than the 2 marks. B says one problem with using biomass in the process shown in figure 4.1 is that the breakdown stage produces a sugar called xylose and ethanoic acid. Yeast cannot use xylose, and ethanoic acid is toxic to yeast. Scientists genetically engineered a type of yeast that can use xylose and ethanoic acid. Figure 4.2 shows the results of one of the trial experiments done by the scientists using their new genetically engineered yeast. So this is the graph over here. It says I describe the results shown in figure 4.2. So first of all, I've written down that the concentrations of the xylose and the ethanoic acid decrease. So over here we can see that the ones with triangles, this is the concentration of ethanol, and the one with circles and lines, this one, you can see over here in the key, this is the concentration of xylose and the ethanoic acid. So this is when they have used their new genetically engineered yeast. We can see that the concentration of xylose or ethanol per decimeter cubed is actually decreasing over here. You can see that it is actually decreasing, both of them. And also you can see that the concentration of the ethanol is increasing. It is increasing the ethanol concentration, and in the same time the concentration of the xylose or the ethanol is decreased, both of them. So first of all, you're going to say that also the concentration of ethanol increases, steep decrease of xylose and ethanoic acid until 62 hours. So over here we can see that in the xylose and the ethanol, we can see ethanoic acid, there's a steep decrease until the hour 62. Over here, a steep decrease means it is decreasing like steadily, like this, like almost like a straight line. So we can see that it is decreasing until hour 62 over here, right over here. This is hour 62. So there was a steady decrease over here. It is almost like a straight line until 62. It's no longer decreasing like before. So this is a point you can write down. Also, there's the steepest change. It is between 6 to 12 hours. So over here we can see that there's the steepest decrease between 6 to 12 hours. From over here you can see that in this hours there was the highest or the steepest decrease. It increased from 0 until 16 over here in just 10 hours. So this is a steep increase. So over here we can say that there's an increase in the ethanol concentration to 44 grams decimeter cubed. And you can also say there is a small change in concentration after or from 62 hours. So you can all say all of this. You can say 3 points of what I have just said and you will get the full mark. Double I, the experiment was done at 30 degrees. The scientists repeated the experiment at 20 degrees. Predict the results that you would expect for the concentration of ethanol. You can say that there will be less ethanol in the same time, take longer to reach same concentration of ethanol. So, if for example, let's say that the ethanol has an optimum temperature of 37 and you put it in like 20, so there will be a lower rate of ethanol because there is less kinetic energy. So this will cause lower amount or lower amount of ethanol to be produced in a specific time. So this is an answer you can write down. Number 5, figure 5.1 shows McLean's cocoa dove, which is found on most of the islands on the southwest of the Pacific Ocean. A says Carker Island is one island where McLean's cocoa dove is found. This species is part of many communities that are adapted to the different habitats on this island. Define the term community. So when it says define the term or define, this means that this is something to be memorized. You can find the terms in the syllabus, you can find it online. So this is to be memorized, all of the populations of different species in an ecosystem. So make sure to always revise on the terms because you can forget them from time to time. Those terms are memorization. Double I, complete the passage with the most appropriate word or phrase. Adaptation is sometimes defined as the process resulting from natural selection. Natural selection is when a group of, for example, animals with a specific characteristic survive because of the environmental pressure on them, by which populations become more suited to their environment over many generations. So this is exactly what natural selection is. B. The highest point on the Carker Island is 1,800 meters above sea level. In 1969, a researcher surveyed the bird species on Carker Island. He recorded the vertical distribution of the birds between sea level and 1,600 meters. In 2013, other researchers repeated the survey. Figure 5.2 shows the ranges of 4 species as recorded in the 2 surveys. Vertical lines represent the range of heights where the birds were seen on the island. So this thick black line indicates that this is from 1969 and the thin line indicates that this is from 2013, as we all can see from the key. So over here we can see that overall in A, in the Myers Goshawk, we can see that in 1969 there's a higher height above the sea level, more than the 2013. The 2013 it was a little bit lower in the height above the sea level. So this is what you're going to say. You're going to say that in 2013 the range it goes downwards to lower altitude. This depends. If you want to talk about 2013, you're going to say in 2013 it went downwards. So let's see B. Over here we can see in the 1969 it was lower than 2013. So you're going to say in 2013 it goes upwards or it extends to higher altitude. So this is what we can see over here. Now over here we can see in C that there is not much of a change. It's almost the same, like they're like very much similar, so there's not much of a change. Now for D, we can see also that there's not much of a change as well. So this was this question. Double I, using the information in figure 5.2, suggest reasons for the change in the range of the island thrush C on Carker Island. So they're talking about C, this one. um, why it might have been this way, why it have been like decreased this way. So you're going to say that there's decrease in populations of the island thrush. This made that this made the range decrease this way in C. So you can also say there's competition between species at higher altitude. This caused the population to decrease. And you can also say that there is increase in populations of other species. You can also say that there is habitat loss or deforestation, which means cutting down the trees, in which some species may have been living there in the trees. So this way they have lost their homes, and increased human activity or tourism, which can cause damage to the habitats. Disease, predation. So this is all like reasons for decrease in populations. You can all write that down. C says small oceanic islands are often inhabited by species of birds that are found nowhere else. Many of these species have decreasing populations and are often endangered. Explain the risk of these species of birds that have decreasing populations. So, if there's decreasing populations of a species, that will cause problems with growth of like a population. So, for example, there will be difficulty finding a mate, which will cause a slower population increase or growth, and inbreeding, and reduced fitness, which means like decreased like genetic pool. There will be less diverse genetics, decrease in genetic variation within each population, or loss of alleles. So, for example, if a bird is, for example, naturally like white, maybe some alleles will be lost. If there is a rare bird, the alleles for it will be lost because there is no genetic variation. And increase in genetic or inherited diseases may spread across this already endangered animal, and it will be more vulnerable to pathogenic or infectious diseases, and it will be unable to adapt to change in environment. So this is all risks of like a small population. 6a figure 6.1 shows diagrams of the circulatory systems of a fish and a mammal. The arrows show the direction of blood flow through the circulatory systems. The first one is actually for the fish, and this is the mammal. I say state the names of the structures labeled Q, T, W, and X. So Q is actually present in the fish. This is the Q one. This is actually the heart. Now for T, it's right here in this place. This is actually the renal vein because it carries the blood out of the kidneys. Now for the W, this is actually the vena cava, which is this one, which is carrying the blood into the heart to get oxygenated. The X is actually right here. This is actually the pulmonary artery. Double I says state the name of structure V on figure 6.1 and describe its function. So V is actually this one. This is actually the septum. The septum is very important because it separates the oxygenated blood with the deoxygenated blood so that they don't mix up. So this is what I've written down, and it also prevents the mixture of both of them. Triple I figure 6.1 shows that fish and mammals have different types of circulation. State why the fish circulation is called a single circulation. So we know that single circulation is when the blood passes through the heart only once in one complete circulation. This means, for example, we have in our mammal heart, the blood goes in and out of the blood of the heart, I mean, 2 times, but in this fish single circulatory system, it only goes in the gills and it moves around into the liver and intestine and muscle until it goes back. It does not go in and out 2 times, it is just 1 time. So this is what is meant by single circulatory system. Now for IV, it says explain the advantages of double circulation of the mammal. So number one, actually, it is efficient supply of blood and oxygen. Double circulation will allow the blood to be oxygenated more efficiently. And it will also allow the efficient removal of carbon dioxide so that the blood is full of oxygen at all times. And also it will lower pressure in the pulmonary artery, It will allow more time for gas exchange because there is more oxygenated blood being pumped in, and there is also a more efficient filtration in the kidneys for excretion, and it allows to maintain a high metabolic rate or rate of respiration. So there is always a high pressure in the pulmonary artery, and there's also a high pressure so that the blood is ensured to be reaching all parts of the body, not just the areas around the heart. B says the blood vessels that supply the heart muscle can become blocked. State the name of the blood vessels that supply the heart muscle. This is actually the coronary arteries. Double I says state one way in which blockages in these blood vessels can be treated. There is two types of treatments. There is the drug treatment, which is by using a drug called aspirin, and there's surgery, which is using stents or angioplasty or heart bypass operation, which means that they will be replacing the heart, all of it. And stents will be like adding a metal, like for example, if you have the artery over here, they will be adding a metal over here. It like moves up and down so that like the blood vessel is opened. C says substances move between blood and tissues at various sites in the circulatory system of mammals. I says oxygen is absorbed into the blood as it passes through the lungs. State the structure in the lungs where oxygen passes into the blood from the air. This is actually alveoli. Double I says state the name or state the site of filtration of blood in the kidneys. This is actually the glomerulus, or you can say nephron. Triple I says state the name of the process in which products of digestion move into the cells and are to become part of cells. This is actually assimilation. IV says state the name of the process in which excess amino acids are broken down in liver cells to produce ammonia. This is deamination. And the last question in the whole exam, state the name of the organ that releases estrogen into the blood. This is the ovary. So this was the end of our exam. Thank you everyone for watching. Make sure to subscribe and like so you can receive more content like this, and good luck with your